Hello, my name is Dr. Jean-Simon Diallo, and I'm the scientific founder and CEO of Virica Biotech. Today, I would like to showcase our proprietary small molecule viral sensitizer platform and discuss some of the applications of the technology and how we collaborate with innovators in the viral medicines industry. Virica Biotech was incorporated in December 2018 and is a spin-out from the Ottawa Hospital Research Institute, where viral sensitizers were first discovered more than a decade ago. The platform originated from work my academic research team was doing to identify ways to improve the growth of oncolytic viruses and make them more efficacious therapies. This initially involved high-throughput screening of small molecule libraries for activity with oncolytic viruses. Since then, we have tested those molecules identified for their activity with many other viruses and viral vectors relevant to gene therapy and the vaccines industry. To date, we've discovered over 100 of these small molecules, and we continue to validate their activity in a wide range of settings. Armed with this platform, Virica's goal is to service the viral medicines industry, which is large and growing, with a lot of promising therapies on the horizon. At the heart of our offering is a solution to a fundamental challenge that faces innovators in the industry: the challenge of overcoming the innate antiviral defense mechanisms present in cells that limit the production and therapeutic efficacy of viral vectors and their derivatives. These ancient and well-preserved immune pathways disrupt development timelines and limit efficacy, posing a serious challenge to the future of the industry. Our small molecule viral sensitizer platform is a solution to this problem, and we believe it will not only accelerate the development of crucial virus-based therapies, but in some cases it may make the difference between a therapy ultimately reaching patients or not. In essence, viral sensitizers, or VSEs for short, attenuate cellular antiviral response, the effects of which are profound both in terms of viral output and efficacy. Most notably, our growing body of data has demonstrated that VSEs can enhance manufacturing yields in some contexts by up to 100-fold. It is easy to appreciate how this can significantly lower production costs for innovators and manufacturers. VSE molecules target diverse molecular pathways and can act synergistically when combined. This allows us to create unique formulations using our high-throughput screening methodologies that can be tailored for most manufacturing platforms. Beyond their manufacturing applications, we also have evidence that certain VSEs could be therapeutically relevant and can be incorporated into viral therapy regimens such as oncolytic viruses to improve their efficacy. In fact, that was our initial objective in developing the technology. The research that we've performed to date suggests that VSEs can be utilized in a broad array of applications that generally fall under three categories. The majority of our validation has been done in a manufacturing context where VSEs can be incorporated to meaningfully increase viral yields and lower production costs. This is an area where we are actively collaborating with several clients. The mechanisms by which VSEs can work also drive the enhanced expression of virus-delivered transgenes. This is relevant, for example, in the production of therapeutic cells, such as in CAR-based cell therapies. Lastly, we have generated indicative data to support the inclusion of VSEs to enhance the efficacy of therapeutic regimens, such as with oncolytic viruses and in vivo gene therapy, and we are seeking strategic partnerships to further explore these possibilities. In the next few sections, I will present some key data illustrating the potential of our technology, and I will wrap things up by providing an overview of how we collaborate with clients across industry and academia. Before I get to specific examples, I would like to spend a few seconds to further illustrate how VSE molecules work. VSEs are chemically defined small molecules that, for example in a manufacturing context, are simply added to cells at or before the time of transfection, transduction, or infection. This schematic depicts how VSEs condition cells by inhibiting cellular antiviral responses so that input plasmid or virus is able to transfect or infect in order for virus to be produced, and in the case of replicating viruses, propagate throughout the cells unencumbered. Ultimately, this leads to an increase in virus production, whether it be for a replicating or non-replicating vector. Viricas Library of viral sensitizers was initially identified through high-throughput screening in the context of oncolytic viruses. Since then, we have conducted over a decade of research to identify over 100 compounds that enhance production in a broad range of manufacturing contexts. We have also created a workhorse library, which is a subset of these compounds, selected for their prior demonstration of activity and the fact that they represent the different chemical classes that comprise our library. This high-throughput expertise also forms the basis of our custom formulation services, where we screen our library to identify VSEs for a specific manufacturing context. An important characteristic of VSEs is how broadly applicable they are to a wide variety of manufacturing contexts, cell substrates, and viruses. This is because the target of VSEs is so fundamental. All cells have antiviral responses, whether they are a manufacturing cell, a therapeutic cell, or a target cell in a patient. Antiviral responses are complex and are the result of the signaling crosstalk between cell substrates and virus products. In the context of manufacturing, we identify VSEs that are relevant for your particular manufacturing process. We have observed that VSEs have activity in a broad range of cells, including cells from different species, transformation status, suspension, adherent, cells grown in serum and serum-free conditions, and even embryonated eggs. VSEs improve production of a broad range of viruses, whether their genomes are DNA or RNA, replicating or non-replicating, or transfection-based. VSEs can be tested directly at your facility, or Virica is able to use our in-house high-throughput methods and expertise to identify a VSE formulation for you. This formulation is integrated into your manufacturing process as a simple media additive. VSEs are added before or at the time of infection or transfection, and there is no need to resupply the compounds throughout your process. VSE concentrations decrease to non-detectable limits in the final product. This is because the compounds are rapidly metabolized by cells and filtered out using traditional purification methods. In consultation with regulatory advisors, we have determined that the inclusion of VSEs in a manufacturing process poses low regulatory risk. To track potential VSE residuals, we have developed analytical methods that sensitively detect VSEs in the final product. Here is an example of our MRM method to detect VSEs in a batch of modified Vaccinia ancara, produced with the assistance of a VSE molecule. Following a standard sucrose cushion-based purification, VSE was not detectable down to the femtomole level. We initially developed our MRM analytical framework due to its stringency, and now work with third-party analytical CROs to validate these methods for client processes and for routine sample analysis. During the initial evaluation phase to identify a formulation, we of course work with research-grade material. However, being relatively simple small molecules, there is really no shortage of CMOs that can manufacture GMP-grade molecule viral sensitizers, and Virica has partnerships in place to provide GMP-grade VSEs when needed by our clients. I'll now present some of the work that we have done to evaluate the potential of VSEs with regards to therapeutic and manufacturing applications. In this slide, we used our high-throughput methodology to identify a multi-VSE formulation that is able to enhance the production of third-generation lentivirus in adherent HEC293 T cells. We chose this process initially because a wide range of academic and even industrial producers are still producing in adherent HEC293T. Depending on the scale, our formulation enhances lentivirus production by 5 to 7-fold, and I should mention here that it is quite adaptable to a variety of different media, including combinations with common additives like sodium butyrate. Here is an example of how a VSE formulation developed for a particular virus strain can be applied to related strains of the same virus. In this case, we developed a formulation that was capable of enhancing the production of a particular strain of MVA. We then evaluated this formulation on 9 genetically distinct strains of MVA and observed variable but consistent enhancements with an average of about 10-fold increase across the different strains. We then took one of the average strains and subsequently took the VSC formulation developed initially at small scale, and then we transferred it without any reoptimization to an iCELLis nano bioreactor. For those who are not familiar with the iCELLis, it is a bioreactor that uses macrocarriers as a scaffold for growing adherent cells over large two-dimensional surfaces, but that take very little space in 3 dimensions. For example, the iCELLis Nano is toaster oven sized, but can accommodate cell growth on surfaces spanning close to 4 meters squared. As MVA is an intracellular virus, we recuperated MVA from supernatants as well as after several rounds of cell lysis, where most of the virus can be recuperated. We observed increased MVA recovery at all stages and in total recuperated approximately 4-fold more virus. To put this in perspective, the amount of virus we recuperated in this context was the equivalent of nearly 8 RC40 roller bottle units. To summarize, this example clearly illustrates 3 important features of the viral sensitizer platform. The first is that a single formulation can be effective across variants of a given virus platform. The second is that it can be transferred and scaled up for use in larger bioreactors. And finally, it can be adapted for use with complementary process intensification strategies, including novel bioreactors for optimal reductions in cost of goods. The data in this slide demonstrates the potential of VSCs not only to enhance the manufacturing of viruses, but also to improve their transduction, for example, in the context of gene therapy. We identified, in this case, single viral sensitizer examples that enhance transgene expression of lentivirus, adenovirus, and adeno-associated virus in a dose-dependent manner. In this context, experiments were done in A549 cancer cells, which are less permissive, for example, than your standard HEC293Ts. And here we were able to observe over 20-fold improvement in transgene expression using, for example, AAV as the vector of choice. As I mentioned earlier, VSCs can be used in vivo for therapeutic applications, and in fact, that was the main intent of developing the technology over a decade ago. So this is an example of a VSC that enhances the therapeutic efficacy of an oncolytic virus in a mouse model of cancer. The antiviral response of tumor cells in the tumor microenvironment is known to be heterogeneous and presents a barrier to efficient oncolytic virus infection of malignant cells. And this is something that's been recognized in the oncolytic virus field for well over a decade. In this case, VSCs are used to inhibit the antiviral responses temporarily, essentially priming the tumor microenvironment for direct infection and spread of the oncolytic virus, without needing to re-engineer the oncolytic virus. The in vivo imaging data to the left shows an acute increase in transgene expression, and this increase correlates to extensions in overall survival when VSCs are incorporated into the treatment regimen, effectively turning an oncolytic virus that is in this case not effective to treat the tumor into one that leads to complete remission. I'd like to wrap things up by discussing how we work with parties in industry and academia who are interested in utilizing our formulation services to develop tailored VSC cocktails to enhance their manufacturing platform. We typically offer 2 ways to collaborate with us. First, clients can opt to test our VSC compounds at their facilities, and we will aid them through our scientific experts who will provide detailed, dedicated guidance on experimental setup, analysis, and formulation development. This is usually performed under a material transfer agreement and assumes that the client has the necessary resources to perform the work using high-throughput methodologies. Alternatively, clients can benefit from our expertise in high-throughput screening and formulation development by outsourcing the work to us under a service agreement. In this scenario, Virica performs the work to identify candidate VSCs that maximally enhance a specific manufacturing platform, and then develop and optimize a formulation of these VSCs that can be transferred to the client for validation at scale and incorporation into their manufacturing process. Ultimately, our goal is to provide compounds in custom formulation to clients on a long-term basis, and our preferred strategy is to do this via supply agreements. We will also support clients with meeting any manufacturing-related regulatory requirements pertaining to the use and incorporation of the VSCs. For clients that wish to explore the utility of our VSEs outside of a manufacturing context, I encourage you to reach out to our business development team through the contact information provided in the final slide or by visiting our website. With that, I thank you for your time and look forward to working with those interested in the future. Thank you.